Lipid management. So the thing about lipids is that whenever you think about lipids, it is always 100% a chemistry problem, because lipids and our bodies do not mix. We have to keep them compartmentalized in order to transport them. And so that's the purpose of lipoproteins. We need to compartmentalize that lipophilic portion of lipids, the fats, and we need to be able to transport them in the circulation, which is mostly water. So just as a review, to start out lipid patterns, in the GI lumen we have triglycerides and cholesterol esters, our primary fats in our diet. These are processed by the pancreatic enzymes, pancreatic lipase and cholesterol esterase, and we result in fatty acids and cholesterol and fatty acid when the cholesterol esters are cleaved. Now these are the base components that are taken up by the intestinal epithelial cells. Remember that these are taken up by the transporter called FATP, fatty acid transport protein. And then this here we're going to talk about a little bit more in pharmacology. This is NPC1L1. It's a Niemann-Pick gene protein. And we'll talk about that in the context of pharmacology. So once we get these components in, the next thing we have to do is reassemble. So we are going to take these fatty acids and we are going to reassemble them into triglycerides. The cholesterol, we will reassemble these into cholesterol esters. And now we have the two primary fats that we are going to be transporting in our circulation. But again, these are lipophilic, so they are incompatible with our blood and our aqueous environment. So we are going to assemble these into the chylomicron. So the chylomicron is simply a specific form of lipoprotein. And the way this is formed is that we have biosynthesis of our apoprotein. In this case, it is ApoB48. So the ApoB gene will express this protein. And then what we're going to do is we are going to add these cholesterol esters and triglyceride units to the ApoB48. And we're going to form a chylomicron. Now, once we do this, the ApoB48, we'll look at the structure of a lipoprotein in general in a few minutes, but the ApoB48 is going to confer enough hydrophilicity so that now the lipoprotein chylomicron is compatible with circulation in the blood. So the chylomicrons enter the circulation after traveling through the lymphatics. They get into the general circulation, and now we have an opportunity to deliver fats to the the tissues. So what we're going to do is we're going to circulate this through the capillaries, and as these are circulating through the capillaries, we are going to be extracting fatty acids. And we'll go through this mechanism of how this works in a few minutes. So this is just a general pattern of how lipids are moving in the blood. We have fatty acids being removed from the chylomicrons, and as this passes through the circulation, we're going to pass this through for approximately 4 to 6 hours. So that means the chylomicron is going to be circulating through the body many, many different times, and that is going to give an opportunity for tissues to be able to take up fatty acids to use them for oxidation. So this is going to go to metabolically active cells, for example, skeletal muscle. Skeletal muscle will use fatty acids for energy. Cardiac muscle. Approximately 80% of the energy generated by cardiac muscle is by fatty acid oxidation. And then, of course, the other one is adipose tissue. So this allows us to extract the fats from our dietary sources of fatty acids and then either use them for energy production or store them for later need. Now, you'll notice this time of 4 to 6 hours. It takes that long to process these chylomicrons because fats do not influence insulin. Okay? Insulin activity, insulin secretion, insulin levels are dictated only by glucose. So that means the fats are not going to induce insulin secretion. What does that mean for uptake of fats by tissues? This is going to be a separate process that is not insulin dependent. Okay? So now, once we're processing this chylomicron and removing all of these fatty acids as we go along, this is going to end up as what's called a chylomicron remnant. And a chylomicron remnant is considered somewhat of a spent particle. And a spent particle, that doesn't mean that it is devoid of fatty acids. It still has fatty acids and cholesterol esters attached to it, but it has gone through the circulation enough to deliver enough fatty acids that now it's time for it to be disassembled and change its form. So at this point of chylomicron remnant, the liver is going to bind this chylomicron and internalize it. Now what the liver is going to do is it's going to take this lipoprotein form of chylomicron remnant. It is going to remove the ApoB48. It is going to disassemble the triglycerides. It is going to remove the cholesterol esters. So what the liver is essentially doing is completely disassembling this form of lipoprotein. Then what the liver is going to do is it's going to synthesize another lipoprotein, another apolipoprotein, and this is called ApoB100. After it does this, it is going to take the cholesterol esters and the triglycerides and reassemble a new lipoprotein. And this structure is now called VLDL. And VLDL stands for very low density lipoprotein. So the purpose of this is the liver is taking our dietary fats and cholesterol and repackaging them into another lipoprotein for long-term needs for fatty acid delivery to tissue. So the chylomicron is what is happening in the fed state. When we enter the fasting state, we still need to deliver fatty acids to our tissues. And so that is the purpose of VLDL. And this can be reflected in the timing. So chylomicrons last only about 4 to 6 hours after a meal. The VLDL can last up to 10 to 12 hours post-meal. So this is the long-acting lipoprotein that is going to circulate in the blood over and over again, providing the availability of fatty acids for tissues. Now what's going to happen to this VLDL over time is that as tissues extract the fatty acids that they need, the particle is going to become smaller. And it's going to become smaller because you're extracting the fats and the particle reduces its size. Eventually, this is going to become, when you take a sufficient amount of fatty acids, this will become an LDL particle. Low density lipoprotein. Now, the LDL particle has had so much fatty acid removed that it still has some fatty acid to it, but this is primarily cholesterol enriched. So as the chylomicrons and VLDLs are circulating in the blood, carrying triglycerides and cholesterol esters, I talked about them, described them removing fatty acids, but I never mentioned cholesterol being extracted from these particles. And that's because these particles, the LDL and chylomicrons, deliver fatty acids, and what is left is cholesterol, and that's the LDL particle. Now, the LDL has two fates. One, the LDL can be taken up by extrahepatic tissue, and of course, this is by the LDL receptor. We'll talk about that a little bit more later. Or the liver can take up the LDL particle itself. Now, the purpose of LDL, and I think all of you know that clinically LDL is quite a challenge, and that's what patients have to be concerned with, because this is where the excess cholesterol comes in that leads to atherosclerosis. But the purpose of LDL is to deliver cholesterol to all cells. So remember, all cells need cholesterol. And so this is the path of how cells get cholesterol. Whatever's left in the circulation is taken up by the liver. Now, what does the liver do with LDL cholesterol? Well, it has two fates. One, the LDL is disassembled, just like chylomicrons are, and then the components of LDL are recycled to make more VLDL and placed back into the circulation. So what this allows cells to do is eventually have a second chance at getting cholesterol if it didn't get it the first time around. And of course, the important clinical route is that the LDL is disassembled and the cholesterol goes into biosynthesis. And so then the biosynthesis is the pathway to rid ourselves of excess cholesterol. Now, the one other lipoprotein that you have to be aware of is HDL, our good cholesterol, as it's called. And HDL stands for high density lipoprotein. Now, we have a relationship we're seeing here between chylomicrons, VLDL, and LDL. The relationship is that they can have their components disassembled and mixed together. HDL is completely separate lipoprotein. It's made by the liver. It has no relationship to the other lipoproteins, but what it's involved in is reverse cholesterol transport. So LDL is the mechanism of delivering cholesterol to extrahepatic tissues, but what if these tissues and cells have taken out the cholesterol they need and they have excess? That's the job of HDL. HDL will take extra cholesterol, scavenge it essentially from cells, and then deliver it to the liver. So already, just from this little bit of information, you can see why clinically you want to raise your HDL levels and reduce your LDL cholesterol levels. Let's reduce the amount of cholesterol going into extrahepatic tissues. Let's increase the amount of lipoprotein that can extract the extra cholesterol and send it to the liver for disposal. Okay, so far? So that's the general pattern of lipoprotein activity. Now, what does a lipoprotein look like? This is what we need to look at to differentiate between these different lipoproteins. So the way I like to draw a lipoprotein is, first of all, let's look at the apoprotein, which is the core. And the core is apolipoprotein B. This core picture looks something like a serpent. So the protein folds into its tertiary conformation when it's synthesized. And while it's folding into this form, we are now adding triglyceride to this. We're adding cholesterol esters, and we're adding free cholesterol. And all of this material is going to be sort of in the core of a lipoprotein. And the lipoprotein is going to be like a serpent in and out of this, essentially like a lipid droplet. The important thing about the lipoprotein are these portions that are exposed on the outside of this lipid droplet, because this is where water and salts can interact in our blood and provide solubility. So the lipoprotein is specifically designed to be able to sequester those lipophilic molecules inside the core and protect them from the aqueous environment of the circulation. So this is what solves our chemistry problem. Now, where do these different names come from? We have chylomicrons, we have VLDL, we have LDL, and we have HDL. Those are the primary lipoproteins we need to be concerned with. The names of these come from the densities by centrifugation. So this is how they were originally discovered. You take the plasma fraction and you centrifuge this, and these individual lipoproteins will separate by density. So if you look at a centrifuge tube, what you'll see is that chylomicrons are very buoyant. And so they are going to essentially float on top of the solution that you're spinning in, centrifuging in. VLDL is very low density lipoprotein. So this is defined by something that is actually a little bit denser than chylomicrons, but it's still very low density, very buoyant. So when you centrifuge this, it's not going to go very far down into the tube because of low density. Now here's LDL. Is not very low density, but it's low density. So it's a little more dense. It's tough on a Friday, right? So it has less buoyancy than VLDL, so it's going to be farther down in the gradient. Where do you think high density is going to be? At the bottom, pretty close, yeah. So this is exactly how these names came about, based on centrifugation through a gradient. And you can actually separate these and fractionate them. This is actually what's done when you're measuring the concentration of these lipids by a clinical lab. Now, what controls the densities of these different products? Well, it's the concentration of triglyceride plus cholesterol. Each one of these particles has approximately the same content of protein. So the protein component of each of these lipoproteins is going to be establishing that core of the lipoprotein. And so the protein concentration in each of these lipoproteins should be about the same, because they should all have a particular lipoprotein. This one is ApoB48. This is ApoB100. This, of course, is derived from VLDL, so it's also ApoB100. This one is ApoA1. And all these are our designations of the core protein that's supporting the skeleton structure. Now, if all the protein is equal, chylomicrons have more lipid component. So if you put lipid in a water solution, does it float or does it sink? It floats. So that means lipids are more buoyant, less dense than water. So the density of these lipoprotein particles is simply dependent on the lipid concentration in the particle itself. So chylomicrons have the most triglyceride and cholesterol. VLDL has a little less triglyceride and cholesterol. LDL is primarily cholesterol. And so what we're doing on these particles is, as you're getting less and less of lipid components, holding protein concentration steady, this is causing the drop in lipid concentration, is causing the increase in density. And that's where these names come from and how they're fractionated differently. HDL is actually a very small particle, and it is almost pure cholesterol. It's almost completely devoid of fatty acids. Okay, so that's where the density and that's where all of these names come from. And I put a table on here for you to take a look at. And this table just looks at these individual lipoproteins based on their densities. And you can see as we go toward HDL, it's more dense. Looking at the protein concentration, percentage by weight, and then take a look at just the triglyceride and cholesterol esters here. And you'll notice that it's that change in ratio of these types of fats to protein which is going to be controlling the density. Okay? Are these specific numbers important to you? No. Okay, so now, those are lipoproteins. So now, in the liver, let's look at the structure of how we construct these lipoproteins in the liver. So, you just saw the chylomicron remnants after they've gone through the circulation a number of passes. These are going to be taken up by the liver, and the liver is going to dissociate this into its components. The liver will take the ApoB48 and send this to the proteasome. The ApoB48 is no longer needed, so it's degraded into amino acids. The triglyceride and cholesterol will be assembled on ApoB100 to form our VLDL particle. Now there's another way to form a VLDL particle. So remember, we're in a postprandial state. We're just after a meal. And so with insulin signaling on the liver, this is going to activate acetyl-CoA carboxylase. And what is that enzyme going to do? Remember that enzyme from yesterday?
Make fat. ACL group.
Make fat, yeah. So this is the committed step in fatty acid synthesis. This is going to take the acetyl-CoA and convert it into malonyl-CoA. So we're in a fed state, postprandial state. So not only do we have lipids and fats coming in from our diet, this is dietary fats, but we also have endogenous synthesis of fatty acids by the liver from the source of glucose. So remember, once our glucose storage capacity is filled, now we're going to take all that glucose and turn it into fatty acids. So the malonyl-CoA and acetyl-CoA are going to form our fatty acids. And now remember, in the liver, once we make fatty acids, we have to get them out of the liver as quickly as possible. Because if we don't, that's when we get steatosis. So the mechanism to get rid of this is through triglyceride synthesis. So we are going to take these fatty acids and we're going to combine them into a triglyceride structure. Now, if we're in a fed state and we have low cholesterol levels so that we're not activating our SREBP signaling system, or are we activating it? Okay, you got to go over that first. So low cholesterol levels will actually activate the SREBP system. And so we're going to increase our HMG-CoA reductase enzyme. And so what we're also going to do in the fed state is make cholesterol, if we need it. So together, the cholesterol and the triglycerides will be mixed with our dietary triglyceride and cholesterol into the VLDL particle for delivery to extrahepatic tissue. So this is the path that is going to prevent us from developing hepatic steatosis. Now, going back to steatosis, when I mentioned about saturating the pathway, this pathway is saturable because it's going to be dependent on the amount of ApoB100. The ApoB100 is not a regulated gene. It's constitutively being made. So there's no way to actually increase the expression level dramatically. So as long as you have ApoB100 around, you can construct your VLDL particles with these triglyceride and cholesterol substrates. So you can see that you have a rate-limiting step here. And once we maximize and exceed this rate-limiting step, now the fatty acids and triglycerides that are being formed in the liver in the fed state start to build up in the liver. So any kind of slowing of assembly of the VLDL particle and secretion into the circulation will lead to the fatty acid buildup and steatosis. So steatosis happens primarily by two routes. One is non-alcoholic, and non-alcoholic steatosis can occur from excessive dietary fats because you're pushing more of these fats into the VLDL synthesis pathway, so you could saturate it from that way. Or it can also happen from excess glucose consumption, because now you're synthesizing more fats from that direction. So thinking about our Western diet that typically is high fat and high glucose carbohydrate, now you've got an increased fat load going into both pathways going into the LDL. And so that's the mechanism of non-alcoholic fatty liver disease. Now, excessive alcohol consumption is going to exacerbate the problem, because alcohol, we'll see about this in about 2 weeks, alcohol is going to produce acetyl-CoA when it's metabolized, and so look what you're doing. You're providing even more substrate for even more fatty acid synthesis. Okay? Now, the pathway for making triglycerides in the liver here from fatty acids is going to be occurring by what is called the phosphatidic acid pathway. This is a different pathway from the pathway that we saw in the intestinal enterocyte. So in the intestinal enterocyte, we saw fatty acids coming in, but remember, when we digested the triglyceride in our diet, we released 2 fatty acids plus a 2-monoglyceride structure. And to remake this triglyceride, to reassemble these 2 fatty acids on the 2-monoglyceride structure to restructure that triglyceride, this is called the monoglyceride pathway. So this is the easy pathway, because all it is involving is taking this 2-monoglyceride and popping these 2 fatty acids back on it to give us our triglyceride. The phosphatidic acid pathway is a little bit more complex, because what we are providing to this pathway are 3 fatty acids. But what we're missing here is a 2-monoglyceride. That's not present in the liver. But we do need the glycerol backbone. And so the glycerol backbone is going to come from glycolysis. Now, again, remember, we're in the fed state, so we have carbohydrates available in the liver to do glycolysis. We're going to take ourselves down to DHAP, dihydroxyacetone phosphate. So remember, this is the triose, the 3-carbon structure following the aldolase cleavage of the 6-carbon fructose. Now normally, this is going to go to glyceraldehyde 3-phosphate and on down in glycolysis. But in this case, we're going to siphon some of this out of the pathway. And we are going to convert this to glycerol 3-phosphate. Dihydroxyacetone phosphate to glycerol 3-phosphate. So it's still the 3-carbon molecule. This is just simply a simple redox reaction to put this, to make the glycerol 3-phosphate. Now, glycerol 3-phosphate looks like this. There's our backbone. So with this backbone structure, now what we're going to do is we're going to add a fatty acid onto each one of these carbons, and we get a triglyceride structure. Okay, so that's a little bit different pathway from the 2-monoglyceride pathway. We'll return again in this next week in this phosphatidic acid pathway, because the alternative to making triglycerides are phospholipids. And we'll look at this next week, phospholipids being the components of our plasma membranes. Okay, questions so far? Yes?
If you eat a meal that has all fats and no glucose, do you get that additional infusion of fatty acids into the LDL?
From dietary sources, or?
Yeah, so you're saying if you eat a meal that has fats and glucose, the glucose will stimulate insulin, which will stimulate acetylcholine carboxylase.
Yes.
If you eat a meal that doesn't have glucose in it, do you get that additional infusion of fatty acids into the
Yes. Not as much, so it's a quantity difference. So think about a fasting glucose level, no spike in glucose, so insulin levels are low. Our insulin, is it go down to 0? No. There's always a little bit of insulin there. And GLUT2 on the liver, I mean, this is a really good question, because now you're looking at the balance of this. The GLUT2 on the surface of liver has a very low affinity for glucose. So it's taking up glucose most rapidly at high glucose levels. When glucose comes back down to fasting level, is GLUT2 inactive? No. Because you're shifting, it's an affinity difference. So GLUT2 activity is significantly reduced, but it's not turned off completely in the fasting state. And the reason for that is because the liver needs energy in the fasting state. And so the liver is going to be using fats for energy, but it also has the capacity of using a little bit of glucose depending on our timing. Yeah. Not a completely realistic clinical scenario, because the only type of diet would be a controlled diet with pure fat, is what we do in the lab. So we can do this with our mice. We can alter and control their diets. So a good example is my breakfast this morning. You know, lots of butter. Dr. Shade's going to come in here and he's going to talk to you about atherosclerosis during the endocrine block. And he's going to bash butter. Oh, he's going to go nuts on butter. So you might see me cringe over in the corner because I just love butter. But on top, what was underneath the butter?
Butter.
Yeah, cinnamon raisin toast underneath, you know? It's under there. So there were some carbs in there which would stimulate the insulin response. Butter underneath, yeah. There's butter underneath the butter and the butter. But, you know, hey, I'm on a statin. I can eat however I want, right?
Says every
Sure, says every patient, right? The importance of compliance with medication and that. My dad's a good example because he had triple bypass surgery after a heart attack at 66 years old. And everybody, including all of his buddies, told him, well, the triple bypass, you're good for about 10 years and then you're finished. Wow. He lived almost 30 years after that triple bypass, and that was because he actually complied with his doctor's orders with his medication control and his diet. So it's, you know, when people say it only lasts 10 years, that's a bunch of baloney. That's a good thing. So, okay, so now, you like the side commentaries I give you, right? Okay, so now, the lipoproteins are delivering fat and cholesterol to cells. So let's look at how the mechanism of how they actually do this. So I'm going to draw a very large blood vessel. So this is the blood. This is our chylomicron and VLDL particles. That are coursing through the blood. And now we have to get these fatty acids to the tissues. So let's say there's skeletal muscle and cardiac muscle that are hungry. Adipose tissue too for storage. So we have to get the fatty acids from these lipoproteins across the endothelial barrier and into these tissues. Now remember that these endothelial cells have pretty secure tight junctions on them to make sure that they're not leaky. So the fats are not going to freely diffuse across these barriers. So what these particular cell types do with the skeletal muscle, cardiac, and adipose is they have to find a way to be able to get those fats to the tissue. So what they're going to do is they are going to synthesize an enzyme called lipoprotein lipase, abbreviated LPL. This enzyme has the same function as pancreatic lipase. Its job is to hydrolyze a fatty acid off of a triglyceride. Now, obviously it can't do this function in the tissue compartment because the chylomicron and VLDL are in the circulation. So this LPL is going to be transported across the endothelial cell layer, and it's going to be placed into the circulation. Now, I'm sure you can already see a problem here. Once something enters the circulation, boom, it's gone. It's flowing away and it's well out of the tissue. So that means the tissue has to find a way to tether it, to hold it in the vasculature in the tissue. So what the endothelial cells will have is a tethering mechanism. And this is a heparan sulfate proteoglycan, abbreviated HSPG. So remember, proteoglycan structures, they have little protein components. This is the protein component here of the proteoglycan, and its job is to simply hold this other structure in place. And this represents long-chain carbohydrates. So the LPL has a very strong affinity for the carbohydrate. So once the LPL is secreted by the tissue into the circulation, it immediately binds the HSPG, and that holds the LPL, even with the blood flow, it holds it within the tissue. Now, as these particles come by the LPL is going to hydrolyze the fatty acids off of the triglyceride. But we have another problem here. These are in the circulation. The circulation is moving very fast. So how can the LPL grab this thing as it's going by? It can't. So what we have now is another lipoprotein that is called ApoE, apolipoprotein E. So the other day when I was mentioning about ApoE, the different isoforms of it, 2, 3, and 4 for Alzheimer's, it's the same ApoE. It has a separate function in the brain, of which nobody really knows yet, but the primary function of ApoE is to act as an address or a locator. And what that means is that this ApoE, also here, has a very high affinity for the HSPG. So by having this bound to the lipoprotein, we now have the lipoprotein itself tethered to the endothelial surface. And again, this is serving the same function as for LPL. It's to hold it in place. Now look what we've done. We've put the enzyme right next to the substrate. And so this here is placing biochemistry on a two-dimensional surface. And that actually has a tremendous effect on the kinetics of this activity. So that we're now able to take and remove those fatty acids efficiently by the lipase activity. Then these fatty acids can cross the tissue barrier and into the cells. Same thing happens with chylomicrons. Chylomicron has the ApoE, it's also tethered, and the fatty acids are hydrolyzed off its triglyceride. Now again, this is fatty acid delivery. So VLDL is going to go through this process, multiple passes in the circulation, and eventually this is going to become an LDL particle, which again is rather devoid of fatty acids, but now it's cholesterol rich. So the cholesterol that was originally in the VLDL remains with the particle, and it's an LDL. Now the LDL can move across this barrier and into tissues, and all cells have the ability to take up LDL. There's a specific transport system that can actually bring LDL across the endothelial barrier. It's the process of transcytosis. Now this is a normal process, and you'll notice a little prelude to Dr. Shade about atherosclerosis is that when LDL moves into tissues, this is an absolutely normal thing, but what if it moves into that vessel? What if it moves into the abluminal surface, the interstitial tissue outside of the coronary artery? Well, this is where we get problems. This is a medial sized artery, and so this point here, the LDL, can become what is considered oxidized. The oxidized LDL is taken up by macrophages. These become foam cells. And you've got an atherosclerotic plaque. So this is comparing the normal function of LDL cholesterol, which is it's an absolutely essential proper function of delivering cholesterol to tissues. But under certain conditions, when the LDL cholesterol molecule lipoprotein is oxidized, and lipids like to be oxidized, now you have the substrate necessary to create an atherosclerotic lesion. Hence why LDL-C levels are supposed to be very low, is to reduce this pathway. Yes?
In the picture on the notes,
Yes.
it has like IDL
Yeah.
on there. What is that?
IDL is called intermediate density lipoprotein. And so when you're removing fats from VLDL, before you get to LDL, IDL is in the middle, intermediate. That's on there for completeness by this author or artist that drew this. But it really has no clinical significance at all because it's a very short-lived particle. Okay, so now, the next step. See, I'm unfolding this little flower for you here because this is rather complicated. We have ApoE associated with these particles. Do you have a burning desire to know where ApoE comes from? Some yes, some no. So let's talk about, did I just ignore it? Everybody said no? So lipoproteins have the ability to communicate with each other. And this gets down to the actual mechanism of how they're working. So when chylomicrons are first made, they are made with ApoB 48. That's it. Nothing else. Just the triglycerides, cholesterol, cholesterol esters, and ApoB 48. And this is called a nascent particle, or nascent chylomicron particle. When VLDL is made, it's made with ApoB 100, and it's put into the circulation as a nascent particle. As a nascent particle, from this lipoprotein lipase mechanism, you can see that the efficiency is very much reduced without the addition of ApoE. So the question is, where does ApoE come from? So HDL has two specific purposes and functions in the circulation. One is reverse cholesterol transport, and we'll talk about that in a second. The second one is it serves as a reservoir for extra lipoproteins. So it's going to have bound to it ApoE, and it's also going to have bound to it ApoC2. Remember that HDL has its own core lipoprotein particle, which is ApoA1. So the ApoA1 is analogous to ApoB48 and ApoB100. But it also has these other apoproteins that are associated with the surface. They're not embedded in the HDL particle. They're actually associated with the surface. And so one of the functions of HDL is to provide ApoE and ApoC2 to complete the chylomicron and VLDL particles. This only happens in the circulation as these particles are bumping into each other. Now, once the chylomicron and VLDL have these two lipoproteins, apoproteins associated with it, now we have a particle that can bind to an HSPG. So here's our HSPG tethered to the endothelial cell. The ApoE is allowing association with that carbohydrate domain. We have LPL coming from the tissues, which is the hydrolytic enzyme to remove the fatty acids. What about ApoC2? ApoC2 is an activator of LPL. So again, this is exactly like the pancreatic lipase. Pancreatic lipase is ineffective in the gut to hydrolyze triglycerides because it needs, what's its accessory protein it needs? Colipase, in order for pancreatic lipase to be active. Under these circumstances, if we didn't have C2, the LPL would have some activity, but with C2, the activity is increased by thousands of fold. And so the particle, the lipoprotein particle itself, is bringing together the components that's necessary to increase the efficiency of fatty acid delivery to the tissues. Now, what happens when the chylomicron remnant is formed after all those passes? And what happens when this particle gets down to an LDL? Be hosting most of the intracellular cholesterol. Now, if there's extra cholesterol there, HDL is going to come swinging by the cell surface, and its job is to extract that extra cholesterol. Now, the problem HDL has is that cholesterol is a very lipophilic molecule. It's embedded in this lipophilic environment of the plasma membrane. So how do you yank this molecule out of its proper environment? Well, that takes energy. If you take it out of this membrane, of the plasma membrane of the cell, and put it into the core of HDL, the energy that it takes to remove it from the cell is equivalent to the energy you're going to gain by putting it into the particle. So that means you have an equal energy, which means that this reaction is reversible. So the HDL could, theoretically, under these conditions, deliver cholesterol, the exact opposite of what you want to do. So HDL has an enzyme, of course. It's called LCAT. And LCAT stands for lecithin, cholesterol, acyl transferase. Lecithin is a fatty acid. And so what this enzyme does is it takes the cholesterol out of the cell and converts it into a cholesterol ester. So this enzyme is associated with HDL. removes the cholesterol, adds a fatty acid to that 3-hydroxyl group, turns it into a cholesterol ester. Now what you've done is you've increased the lipophilicity of cholesterol. So once it gets into this particle, there's no going back. You've increased the solubility by this reaction for the particle. And so that makes sure that this reaction is not reversible. Now, as it turns out, LCAT is a very important enzyme for you to remember. And the reason for that is, do you remember the other CAT enzyme we saw?
ACAT.
ACAT. Do you remember what ACAT does?
It seals fatty acid for prevent reverse transport.
Yeah, acyl cholesterol, acyl transport. ACAT. This enzyme is located inside intestinal cells plus hepatocytes. And its job is to take cholesterol esters and add them to either a chylomicron, if you're an intestinal cell, or VLDL. Now this is where the step 1 exam gets mean. They want you to be able to differentiate between these two enzymes and their location, their purpose, and their function. Is it easy to get those two confused? You bet it is. So this is the difference, okay? Okay, so now the HDL, once you have the HDL particle loaded up with cholesterol, this is going to go to the liver. The liver extracts the cholesterol and it takes the HDL particle and sends it back into the circulation for additional rounds of reverse cholesterol transport. Okay? The cholesterol that the liver has extracted can be used to generate new VLDL particles or disposal as a bile salt. Okay, so let me backtrack 1 second and go back to LPL, because I forgot about this. This is an extremely rare mutation on LPL. This particular mutation, I think, is most prevalent in Asian populations, and I think the frequency is the highest in Japanese population. But mutations in LPL can occur in individuals, and if you have a mutation in LPL that eliminates it, there's a loss of function mutation, what is the result going to be for your patient?
Can't uptake fats, you just have too much in the blood.
Atherosclerosis, right? Because now here, you still can make VLDL, you can still do the chylomicron synthesis, you can still put it in the circulation, but now you can't extract the fats. So the concentration of these lipids increases in the blood, and this mutation leads to hyperlipidemia. Excessive hyperlipidemia, it's huge. So we're talking on the order of triglycerides, you're supposed to be like 120 mg per deciliter. We're talking about anywhere from 1,500 to 3,000 mg per deciliter. So obviously a mutation like this is going to be an early atherosclerotic disease. Now the reason I bring that up is because of LDL. So the LDL receptor if this is the LDL receptor, and this is LDL, the purpose of this receptor is to internalize the LDL into intracellular vesicles. What happens at this point is that these vesicles become acidified. Lysosomal vesicles become acidified, and the LDL dissociates from the receptor, which will give us two compartments, one with LDL in it, the other one with the unassociated receptor. So this particular vesicle is called a recycling vesicle. It's simply because what we're doing is we're separating the ligand from the receptor and returning the receptor unbound to the cell surface to undergo additional rounds of internalization to keep taking up LDL. This goes to lysosomes, where the LDL particle is broken up, and then the fatty acids that are left, and mostly cholesterol, are able to be taken up and used by the cell. Now, this is what normally happens. Familial hypercholesterolemia. We had a medical student here that had this about 10 years ago. I was talking about this, and after class he said, "Yep, I got that." Oh God, sorry dude. So what this is a mutation in the LDL receptor. The first one that was ever identified by Brown and Goldstein at the University of Texas Health Science Center in Dallas, Southwestern. And they identified this because they had patients, several of them, that tended to be approximately 40-year-old males. And these 40-year-old males had a heart attack, and many of them died. 40-year-old male having a heart attack, what's up with that? So what they did is they started looking at these individuals, and they found that their LDL-C levels, LDL cholesterol levels, were much greater than 1,000 mg per deciliter, which means that their circulating LDL cholesterol is very, very high. So the reason they found out it was high is because these individuals have a mutation in their cytoplasmic tails of their LDL receptor. This mutation, the LDL can bind just perfectly fine, but the internalization event is prevented. So as you can imagine, with LDL cholesterol, you eventually saturate all the receptors, but if you can't internalize the receptor and unload your cargo, you cannot return the empty receptor. So the LDL-C levels start to really rise on these individuals. This is classical familial hypercholesterolemia. Now, since then, there's been other mutations in the LDL receptor that have been identified. Some others in the cytoplasmic tail that prevent internalization, and others in the binding site for LDL for the receptor, so that the ligand can no longer bind to the receptor, and the outcome, clinical presentation, is the same.